Welcome back, folks. This is video lecture on Chapter 7, Part A, looking at sensors, how we sense the world around us. This video only concerns Section A of the topics. Another video will address the second half of the chapter. As the words suggest, sensory receptors are receptors in the body which can sense the world around us, as well as the world within. These sensory receptors are located at the very peripheral end of the afferent neurons that carry information towards the central nervous system. A stimulus is the trigger enabling these cells to generate electrical activity. The degree of this electrical activity is known as a graded potential because it can have values that are anywhere on the scale. These graded potentials have a name called a receptor potential. And if the receptor potential is great enough, it will activate an action potential in either the same neuron or in an adjacent neuron. This information is usually relayed to the central nervous system so that decisions can be made. There are 5 major classes of sensory receptors throughout the body. We have mechanoreceptors, which respond to mechanical distortion. We have thermoreceptors, which respond to temperature. We have photoreceptors that respond to light energy. We have chemoreceptors that respond to all kinds of chemical stimulus. And then we have nociceptors, which are pain receptors. Regardless of the class, these sensory receptors fall into 2 architectural relationships. Either the sensor is the neuron that carries information to the central nervous system, or it's a specialized receptor cell that talks to another cell, which is then the neuron that conveys information to the central nervous system. In a few specialized cases, we could have a number of cells before the neuron. Regardless, the direction of information transmittance is always from the stimulus to the receptor, and then from the receptor to the central nervous system. As an example, we'll look at the former case and show you that if we use probes to detect the type of stimulus or the electrical activity, we find that at different lengths of the receptor, the signal has different profiles. The first thing to notice is that the stimulus has to have a certain amplitude, a certain magnitude. When the magnitude is small and it's barely detectable, then it may generate some electrical activity in the first segment of the cell, but that does not lead to any action potentials. If the stimulus is increased in amplitude, then the corresponding increase in the graded potential does result in a firing of action potentials with a low frequency. The frequency is the number of action potentials per unit time. And as you travel to the far reaches of that neuron, the action potentials, being all or nothing, continue to be detected. As the amplitude of the stimulus increases further, it does increase the graded potential. That is going to lead to action potentials, but at a very fast frequency. So the frequency at which the action potentials are firing is now much faster than these, although they are the same magnitude. And this will again be detected in the far reaches of that neuron. At the axon terminal, in the first column, there is no release of neurotransmitter. In the middle column, there's some release of neurotransmitter, and in the final column, there's a substantial release of neurotransmitter. And that information corresponds to the strength of the stimulus detected at the front end. Figure 7.3 compares 2 types of receptors. Some are called slow adapting, others are called rapidly adapting. So the rapidly adapting profile is given on top, and the slow adapting profile is given below in both brown and blue. The teal line corresponds to the stimulus being applied, kept on, and then being removed. The 2 classes of receptors produce their own characteristic responses. With the rapidly adapting receptor type. Stimulus causes a graded potential to be generated within that cell, but that graded potential only lasts a specific period of time, even though the stimulus is still applied. So the graded potential dies off, and it causes a rapid firing of action potentials, which also die down in frequency, and then at a certain point there is no more action potentials released. The stimulus can be kept on, and once the stimulus is removed, the change in stimulus is detected by a graded potential, and that fires further action potentials. An example of this will be having your underwear being first applied to your body, and then for the rest of the day you don't notice your underwear until you remove the underwear in the evening. However, with the slow adapting tonic receptors, the action potentials continue as long as a stimulus is applied, but their frequency begins to diminish. The most important concept is the word coding when it comes to sensory perception. Coding is the conversion of a stimulus energy into a signal that conveys the relevant sensory information to the central nervous system. The central nervous system only has two parameters that it can utilize every time along the electrical neurons. The first, as we've seen, is the frequency of the action potentials, and the second is the amplitude of the graded potentials. Using a water analogy always helps. In this regard, we're looking at a stone being thrown into a pond, and it generates ripples. The frequency would be how close together ripples are in a pond, and the amplitude will be the height of those ripples. With respect to the stimulus itself, its characteristics would include the type of input it represents, its intensity, and the location of the body that it affects. All those will be taken into consideration when the information reaches the central nervous system. Figure 7.4 shows a prototypical neuron with its receptive field embedded, in this case, in the skin. So the coverage of the peripheral terminals, the dendrites, cannot relay which of these branches is being activated. All this cell can do is tell you the intensity of activation along all these branches combined. The receptive field represents the area that the dendrites cover. Additional information available from this figure is that the cell body of the receptive afferent neurons is normally located very close to the central nervous system, but their processes can be extremely long. The modality of a stimulus is simply the type of energy that it carries. So we have examples of heat energy. We have examples of cold energy. We have examples of pressure waves. We have sound, and we have light. These are the most common modalities that the body can detect. The various receptors that have evolved have become specialized to detect a particular type of modality. The receptors are no more than specialized skin cells in most cases. Figure 7.5 nicely conveys the concept of touch and the perception of that touch as action potentials by the central nervous system. Once again, there are three parts to this figure. The bottom part is the pressure applied with the glass probe, and the middle illustrates which portions of the dendrites are activated, and the top measures the electrical activity, the action potentials generated in those neurons. When there's very light touch, in this case less than 60 millimeters of mercury, the indentation in the skin is not sufficient to trigger any action potentials in this neuron. A more aggressive touch, about 60 millimeters of mercury, causes one dendrite to begin to be pressured, and that fires a frequency of action potentials. When the pressure is increased to about 100 millimeters of mercury, the indentation in the skin causes a physical deformation of two dendrites, and their combined effect is a higher frequency of action potentials traveling down the neuron. And as the pressure increases, we recruit more and more dendrites, and therefore the frequency at which the action potentials are fired proportionately increases. So this signal, as it's received by the brain or the central nervous system, would be coded. So this tells you there's a large pressure on this neuron. This tells you there's a mild pressure. The size of the receptive field for any particular sensory neuron is proportional to its importance to the body's survival. So some areas are covered with neurons that are tightly packed, such as the lips, and other parts of your body are covered with neurons that have large receptive fields, such as the middle of your back. So each of these has a very different profile when it comes to its value in detecting and sensing danger. The body simply does not have enough room, enough width to carry all the cables at a density equal to that of the lips. Thus, some regions are sacrificed and given sensory cells with large receptive fields. The point at which the stimulus impacts the receptive field of a neuron is irrelevant because there's no way the cell can tell which one of these dendrites is being stimulated. However, we must take into consideration the arrangement of neighboring neurons or sensory cells. They also have receptive fields, and when something stimulates one cell, it may automatically overlap and stimulate neighboring cells. One might calculate that the action potentials generating in neurons A, B, and C would follow this profile here, with the majority of stimulation taking place in neuron B, but neurons A and C stimulated about 1/3 that of neuron B because of the number of dendrites involved. Fortunately, in this case, it's going to be pretty difficult for the body to detect the exact point at which the stimulus is being applied. Amazingly, evolution has allowed the development of something known as lateral inhibition. In this process, neighboring neurons are somehow able to reduce their action potentials at the expense of giving the central cell, cell B, a maximum response rate. The methodology behind this is known as lateral inhibition. The mechanism involves reducing the input from neighboring peripheral regions, therefore focusing the brain's attention on the exact sensory point. The mechanism has now been discovered how this actually happens, with figure 7.9 trying to relay that to us. If we start at the bottom of the figure, we can see that under normal circumstances, if you go back to here, we would expect that neuron A will be firing at 1/3 the frequency of neuron B. And we see that in the coding of the frequency here. However, when the information is perceived by the central nervous system, one can see that the coding ratio has been substantially altered in favor of more signal arriving from the center cell than the peripheral cells. And this is because of lateral inhibition. These interneurons, neurons between the processes of these neurons, they, when activated, will decrease the action potential generated in post-synaptic cells. The result is some inhibition of the frequency of action potentials in cell B, but not to the same degree as cell A and cell C. Thus, as far as the central nervous system is concerned, cell B is the one that's been stimulated, and cell A and cell C are just being tinkled. Figure 10 relays that information to us. The purple line represents the initial stimulus at the tips, and the teal line corresponds to what's received in the central nervous system, with the focus clearly narrowing down the area of stimulus. The red interneurons play a very, very important role in achieving this outcome. Although we lack a complete understanding of how the central nervous system functions, we're beginning to understand various architectural aspects and regulation. One thing emerging from all this research is that the signals, as they are passed from the periphery to the central nervous system, they go through extensive modification before they reach the higher levels of the central nervous system. These modifications can take the form of inhibition or excitation. The exact interplay is quite complex and can result from ascending neurons traveling together, descending pathways, neurons coming down from the higher centers, and by synapses on the axon terminals on these neurons, or indirectly by interneurons. The classic example of this being the pain pathways. If these were operating at 100%, then it would not be possible for us to detect further pain. Therefore, after the initial stimulus, these are dulled down, the action potential coding frequency is reduced, giving us the option of increasing that frequency should more pain arrive. Figure 7.11 tries to convey that relationship. The green neurons are excitatory and the red neurons are inhibitory. Another architectural outcome is that of divergence versus convergence. In divergence, we have one neuron that interacts with multiple neurons once it enters the central nervous system. So information from this one neuron can be conveyed to multiple centers in the brain. In convergence, multiple neurons converge on a set of interneurons in the central nervous system. Of the two, which one conveys more information? Well, the former is more informative, but sometimes there's not enough space to have a one on multiple relationship, and the result is that multiple neurons are then going to interface with interneurons in a complex fashion such as this. Information carried by these sensory pathways has more opportunity to diversify if it connects to multiple neurons on its pathway. Thus, evolution has equipped our bodies with chains of 3 or more neurons connected by synapses that form the ascending pathways as the information enters the central nervous system. In some cases, there are specific pathways that carry unique information from a particular modality. In other cases, non-specific pathways carry information from combined modalities or from combined locations within the body. Regardless of which of these pathways is used to arrive, once the information arrives in the primary cortical receiving areas associated with each sense, then further interconnections are made into association areas in the cerebral cortex, where complex integration occurs in a yet unknown manner. Although we are not going to study the brain in this course, it's important to have a general understanding that the brain itself is organized into centers, and those centers are geographically mappable onto the brain. Figure 7.14 shows a specific and a non-specific pathway. So in this case, we have temperature being sensed by these neurons and touch being sensed by these neurons. And the connections up into the brain are along unique paths. So the brain can sense temperature independently of touch. In other areas, the two neurons, the sensory neurons for touch and temperature, interconnect to a single neuron. And in this case, the upper areas of the central nervous system, the higher centers, are unable to tell whether there's touch or temperature. They know there's going to be one or the other, but they can't discriminate between the two. So these are non-specific ascending pathways, and these are specific ascending pathways. Sometimes there's simply not enough space in these tubes to carry information in a one-on-one relationship. So priority is given to different areas of the body where these two would be very important to discriminate. It appears that the wiring of the brain has some geographical orientation beyond simply localization. The primary somatosensory cortex relies upon various cortical association areas for proper processing of sensory information. Also to modify that information based on many factors, including emotion and motivation, arousal, attention, memory, and language. Thus, a person who's depressed at one time may respond completely different when they're not depressed. Factors that can affect perception are listed in this slide. The most important appear to be our emotions, our personalities, and our past experiences. There's also much sensory information that is not sent to the conscious part of our brain. For instance, we're not aware of our blood pressure on a continuous basis. In addition, we have lack of receptors for certain stimuli. We may have damaged neural pathways. We may be taking drugs which interfere with the processing of information, or have other types of pathophysiology either caused by genetic or non-genetic factors. Table 7.1 does a good job of presenting a summary of the stimulus feature and the processing of the stimulus thereafter. Sensation coming from various parts of the body, including the skin, the skeletal muscles, bones, tendons, and joints, is referenced as somatic sensation, and it's initiated by a variety of sensory receptors collectively known as the somatic receptors. These receptors give us the ability to sense touch and pressure. They allow us to be aware of our body's position and their movement. They also allow us to determine temperature, pain, and itch. Our skin is loaded with many different types of sensors simply because that skin is in contact with the external world. Information from that environment is fundamental in our survival, therefore we need to detect it. Please learn the different names of the different receptors and what they detect. Pain is one of the more important senses that we have because it alerts us to danger. When pain is detected, afferent neurons carry that information toward the central nervous system, where certain neurotransmitters like substance P and glutamate are utilized to communicate to the ascending pathways to the brain. Unfortunately, we also have something known as referred pain, where different neurons are interconnected in a way that makes it hard for us to know exactly where the pain is coming from. In this figure, we reveal that there's convergence taking place at this point between two neural pathways. The first emanating from the heart and the second emanating from the skin in the armpit or the arm itself on the left-hand side of the body. When pain is received in the brain from the heart, it feels like we have pain in the arm. So heart attacks are normally felt as arm pain rather than pain directly in the chest. Referred pain has been mapped. Any pain, for instance, in the neck is convergence from neurons carrying information from this region as well as from the lungs and the diaphragm. Thus, the correlation between surface pain and visceral pain may have been evolutionary more useful than direct pain coming from within the body. Certain pain lasts longer than the stimulus, mainly to warn our body to be cautious with that damaged area. In addition, hyperalgesia is an increased sensitivity to pain stimulus. Incidentally, a paper released in July 2019 has shown that in invertebrates, such as the fruit fly, hyperalgesia exists too, in that the researchers were able to show that once they damaged one leg of a fly, it was very cautious and fearful for future damage to other limbs. Amazing. Pain management techniques rely on our understanding of the physiology, sensory neurons, and their interconnections. Analgesia is the suppression of pain without affecting our consciousness. We have several avenues of action, including electrical stimulation, using pharmaceutical drugs over the counter, or by investigating endogenous opioids produced by the neurons themselves. Acupuncture seems to be a useful technique in stimulating the release of these endogenous opioids, and a technique called TENS, which is transcutaneous electrical nerve stimulation, can also be used. The purpose of these various techniques is to inhibit the activation of the ascending pathways to the brain. So if the brain doesn't sense any pain, then there will be no pain. Itch used to be thought of as a failure in some neuron communication, but more recently it's now been studied as a somatic sensation in its own right, distinct from other mechanisms such as pain pathways. Two types of itch are now recognized. One is acute itch, and that involves a pathway that appears to be completely separate to chronic itch, utilizing different types of receptors. The other thing we're trying to understand is why the pathways leading from the input from one side of the body cross over and feed the other side of the brain. It may have something to do with injury and functionality. Regardless, there appear to be two conduits moving up and down the central nervous system. The first is called the anterolateral system, and that involves the front part of your spinal cord. And the other is the dorsal system, and that involves the back end of your spinal cord. With both, switchover does take place, but with the anterolateral system, it occurs at the local level, at the point at which the afferent neurons enter the central nervous system. In contrast, the dorsal system is over at the level of the brainstem. One is associated with one type of modal input and the other with the others. Temperature receptors and pain receptors use the anterolateral system and cross over immediately, whereas movement, limb position, fine touch, and pressure use the dorsal pathway and cross over in the brainstem. The amount of room, the amount of space given to different body parts with reference to the density of receptors feeding the higher centers of the brain is proportional to your survival needs. The face, especially the lips and the area close to them, your fingers, your hands, and your genitals are given priority with respect to bandwidth, which translates into space. The mapping of these locations to the somatosensory cortex is illustrated in figure 7.21. In the next video, we'll look at each of these senses and how they operate.